Hi, everyone. Happy Movember, and welcome to another edition of our MGS Biolinks Breathe and Learn. I'm Steve McKen from MGS Biolinks, and it's my privilege today to introduce you to our guest. Matthew Cuvato is the head of product development at ProtonDx, and he's joining us to share with you how you can cut your viral RNA and DNA extraction times down significantly with their new product, SmartLid from ProtonDx. So with that, I will pass it off to Matthew. Thanks so much for joining.
All right, welcome everyone. For those of you who don't know, ProtonDx is a sample preparation and diagnostics company. But importantly, what allows us to be a high quality diagnostic company is our high quality sample preparation and that side of the business. And that's what we're going to be talking to you about today. We are a company full of researchers and molecular biologists and people who have spent years in the lab working with the competition's extraction and sample preparation kits. And we're really tired of the long, complex, and restrictive protocols. And so we wanted to design something that was better, and that's how we came up with SmartLid. So I'll go through a bit about the product in this presentation, and then I'll give a short demo and we can open it up for questions. So SmartLid in its current form is a viral DNA and RNA extraction kit that extracts DNA and RNA from cell-free fluids, or mostly cell-free fluids. It's a 50-pack, and it includes everything you need to get a high yield and really easy to use extraction out of it. And it has everything you need in the box, meaning no equipment required, no electricity, as you'll see in a little bit. So SmartLid is special because it centers around two key components. One of them is the SmartLid itself, which you can see on the left and the right. It's a disposable lid that fits into your extraction tube and has a removable magnet, which is this green part here, that collects magnetic beads or magnetic nanoparticles out of the solution and onto the lid. And you can see that on the right, this little mound of black particles. And that allows you to easily transfer the magnetic particles through a series of sample extraction steps. And the other part of what makes that so easy and so quick are the magnetic beads that are exclusive to our company. I challenge you to find a faster collecting magnetic bead anywhere on the market. And you'll see that in a bit. It's something that really shows a lot better in video or in person. And this is just illustrating that the beads really just disappear from the solution when collecting onto the magnet and to the SmartLid in a matter of seconds. And that's something that I'll show you in a second. Our protocol is able to achieve comparable or better results to market-leading competition across DNA and RNA, whether it's from viral particles, as shown in the data on the left, or just from recovering nucleic acids directly from samples, as is shown on the right. But really importantly, it does that in a fraction of the time, with the average protocol length less than 10 minutes. And that's whether you're extracting 1 sample or 12 samples, thanks to some of the accessories that we offer, and I'll show you. And as you can see here on the screen. So I think I might as well jump straight to the demo. And if you have any questions about the presentation or the demo, we can hit them afterwards. So I'm going to go through what's in the box first, and then go through an example extraction explaining sort of what happens. And for anyone who's not familiar with magnetic bead extraction, I'll go through some high-level concepts, and then I'll show you how we process multiple samples at a time. So this is the box. As I said, everything you need is in here, minus ethanol and isopropanol, which you supply in your labs like any other kit. So what we have here is just the IFU, and this here links you to a set of really good training videos on the product, and we think that that really helps understand the product if you're doing it for the first time. We have 50 SmartLids, so these are disposable, and these are what you use 1 per extraction of. You have the tubes that specially fit SmartLid and will contain all of your buffers. We have a set of 12 magnetic keys, and there's only 12 because these are reusable. So you can reuse these from extraction to extraction over a long period of time, and allow you to perform up to 12 samples at a time. If you want to do more, you can buy another starter kit, as the refill kits don't come with the magnets anymore. And then the last thing are our custom developed buffers designed to give you a really pure and high yield sample when working with SmartLid. So you have your lysis and binding buffer, you'll have your magnetic beads, you have your wash buffer, which you add ethanol to, and you'll have your elution buffer. And now just going briefly through the protocol. So this is one of our accessories, the SmartLid rack, and you can see it here in 2 parts that come together to allow you to do 12 extractions at a time if you want to. And so just going through the process will get you a little bit closer. There we go. You essentially only have 3 quick steps. So you have your lysis step, where you're also binding all the nucleic acids that you're releasing from your sample, from the viral capsids, from the cells, into binding them onto the silica that's coated around the magnetic particles. You then take the beads from the first tube to the second tube, where you wash away everything you don't want. So you don't want the lysis buffer, you don't want proteins and lipids and all this in your final elution. And then you'll dry the beads briefly and then put them into your elution tube, which concentrates the nucleic acids and acts as a really pure sample to then go into downstream analysis, whether it's LAMP or PCR or digital PCR. So I'll show you the collection, which is the cool part right here. So as you can see in the bottom of this tube, we have the magnetic beads in your lysis and binding buffer. And if I give it a quick shake, the liquid turns a really dark black, so it's pretty clear where the beads are. And if I just open the tube and put in SmartLid with the key, the magnetic key inserted, and again, this just twists to lock and gets the magnet really close to the liquid, you'll see a quick collection onto the tube. And I'll do that one more time just in case that was too quick with any lag that's happening, and get that very close to the camera. Here we go. And the beads are nearly completely gone. So after, myself having years of experience with other kits, that was a huge improvement already, just the speed of collection. And you can imagine how much that speeds up your process. So you're not waiting anymore for minutes to collect at each step. You're just waiting a few seconds. And you can see the beads are now collected on the underside of SmartLid in this little bowl. And now SmartLid has these tabs here, so you can grab SmartLid, you can put it on a table, you can put it on the stand, and you're not going to let these beads contact anything. So when you're ready to put it into the next tube, you just pop SmartLid in, and this time you're going to remove the magnet and give it a quick shake, and the beads drop into the washing buffer. And so at this stage, you can just shake it up, washing and rinsing away all the proteins and lipids. Pop the magnet back in, and again, a really quick collection of all the particles, and your liquid's clear of the beads. As I mentioned, you're going to dry the beads. You want any ethanol that's in that washing buffer to evaporate. So you can just set that down for 30 seconds to a minute. I won't wait that long now, but then you can transfer the beads right into your elution buffer. And here, the elution buffer is going to separate all of your nucleic acids that are on the beads from the beads themselves and drop them into solution. And again, mixing is as simple as gently shaking at this stage. You can, if you want, vortex, of course. And then popping in the magnet to collect the beads again. And then that's about it. You have your magnetic beads on the SmartLid. You can take off the key and save that for later extraction, throw away the SmartLid, and you have your purified and concentrated nucleic acids in this final tube. And that can then go down to your any analysis that you want on that. And just, I'll show you briefly how that works. The same process when you're doing multiple samples at a time, you can bring in a rack like this. You can easily number all your smart lids. They're designed with a nice texture here to write on. And you can process using this tool that we created, the SmartLid Shaker, for up to 12 samples at a time. And the shaker is numbered so you don't lose track of what extraction you're on. So you can pop any number you're doing into the shaker, lock it on that side, and then shake all the samples at once. You can see all the beads resuspending there. And when you want to collect all the beads, you can also just pop in the magnetic keys one at a time into the smart lids that are in the shaker and collect all the beads at once. And hopefully you can see that just as fast as when you're doing one, but now I've done 6 at a time, and you can do 12 at a time. And at that point, all the beads are collected, and you can pop them right back in the rack, transfer them onto the next tube, and continue your process. And hopefully you can see why this is so much quicker than kits and protocols you're probably used to.